There can also be genetic exchanges of material between living bacterial cells through a process known as conjugation. In this video, one of the cells has a plasmid. This is an extra chromosomal loop of DNA. And this plasmid encodes genes for its own transfer. The cell which possesses the plasmid forms a pilus, which then binds and attracts another cell. And then the plasmid, after replicating itself, passes from the first cell into the second cell. And so that both of these cells now possess this F plasmid. Both have gone from being F minus to F plus. If this plasmid possessed a gene encoding a trait, it would be known as an F prime plasmid. And in the following example, there is a plasmid which encodes a gene for antibiotic resistance. So in this example, we put two different strains of bacteria on lateral halves of a petri dish, which we use a marker to split the petri dish into half. And the plates which simply have luria broth agar, in other words, which are just food, they should allow for the growth of each of these two strains. We can call them strains X and Y. We notice that each of these two strains can grow in the presence of one antibiotic, but neither can grow in the presence of both. So strain X does not grow on the plate which possesses ampicillin, but can develop on the plate which possesses streptomycin. In contrast, strain Y grows on the plate with ampicillin, but does not develop on the plate with streptomycin. So each of these is resistant to one antibiotic, but not both. Two plates are used which possess both ampicillin and streptomycin. Now, if the bacteria are simply placed on either side of a plate such as this, then none of them are expected to survive, since neither of these two strains is resistant to both antibiotics. The plate possesses at least one antibiotic which will kill the cells. However, the second plate is converted into a mating plate. In it, the bacteria are placed on two sides of the dish, but then in the center, they are mixed as a loop draws bacteria from one strain towards the other and then back, giving them the chance to undergo conjugation. If one strain converts or confers a plasmid with the resistance to one antibiotic into a cell which in its nuclear genes has a gene for resistance to the second antibiotic, then there will be bacteria which are resistant to both antibiotics, which would be able to thrive on this plate and form colonies in the center region of the plate where conjugation was allowed to occur.